Hi, this is Jay, and I'm gonna be presenting the paper that I've read. My paper is Caffeine Controls Glutamatergic Synaptic Transmission and Pyramidal Neuroexcitability in Human Neocortex. So, caffeine is one of the most used chemicals in our lives. For example, we often start the day with 1 or 2 cups of coffee. However, have you ever wondered how caffeine works in our brain? Obviously, caffeine will work in the same way that we learned in class. Caffeine binds to the receptor and antagonizes the receptor and prevents the receptor from accepting other neurotransmitters. So the big question of the research is how does caffeine influences the human brain in what way? The goal here is to investigate the effects of caffeine of the human brain. This investigation is important since the impacts of caffeine in a human brain was not investigated but in rodents. So adenosine, the chemical in our brain, is known to bind with A1B. So first of all, they check if the human brain they collected has abundant amounts of A1B. This is important because if there is no A1B present, they cannot assess the effect of caffeine in this receptor or brain in general. They also compare the ability of caffeine on another antagonist, HDPCPX, to bind with this receptor. There are enough amounts of A1R, and this experiment demonstrates that caffeine can act as an A1B antagonist like HDPCPX. Second, they checked if adenosine, caffeine, and HDPCPX impacted pyramidal neurons' intrinsic membrane properties, such as input resistance, resting membrane potential, voltage sag, and wheelbase, which is the current injection needed before firing off the first action potential. The result shows that in adenosine, resting membrane potential did not decrease. Input resistance decreased and wheelbase increased. This is due to the fact that A1R activates when adenosine binds to the receptor. In other words, since the receptor has activated, this intrinsic membrane properties is affected. In HDPCPX, there were no effects in A1R itself. Since caffeine acts as an antagonist just as HDPCPX, there were no effects in A1R either. Then they tested the effects of adenosine with caffeine present. Let's think about the person who drinks a moderate amount of coffee or caffeine. Adenosine will always be present in that person's brain, and drinking coffee will impact him by adding caffeine in their brain, right? So this step is to check how caffeine affects the human brain. So it was observed that the effects of adenosine in A1B were affected when presenting with caffeine. Authors described that caffeine partially blocks the effects of adenosine. This can explain that caffeine can interfere with the results of adenosine on the intrinsic membrane properties of pyramidal neurons. Lastly, they tested the effects of caffeine on genetic transmission. They used the amplitude of excitatory events and frequency of excitatory events in glutamatergic genetic transmission. Adenosine itself decreases both amplitude and frequency. HTPCPS itself increases the frequency but not amplitude. It did not change. So caffeine, on the other hand, did not change at all. Then they tried to add two factors, like two chemicals, to test if it changes the genetic transmission. Both amplitude and frequency were blunted by HTPCPX and when presented with adenosine. According to the authors, this suggests that HTPCPX will impact the activity of presynaptic A1R to inhibit glutamate release. On the other hand, only amplitude decreases when caffeine is introduced with adenosine. Authors suggest that caffeine might impact the postsynaptic A1R. So in summary, moderate concentration of caffeine counteract the effects of adenosine inhibitory effects. This is because caffeine acts as an antagonist in A1R. In the third experiment, we can see that caffeine affects the postsynaptic A1R and affects the amplitude of the excitatory events of adenosine. On the second experiment, caffeine binds to A1R and affects the pyramidal neuron's intrinsic membrane properties. Briefly speaking, caffeine affects the human neocortex by binding to A1R and interferes with adenosine to bind to this receptor. Also, it acts in a postsynaptic A1R and affects synaptic transmission. Thank you for watching this video.